Hello guys, today my topic will be on acid-base balance. This is a very complex topic and very hard to grasp, so I've combined a lot of electrolyte disturbances, what is acid, what is base, how do we really go about this. If you have any doubts or questions, please ask me. Okay, so acid-base balance. Acids can be divided into two types, one is volatile and one is non-volatile. What is the key difference? Volatile acids are acids that can easily be expired through your lungs, which includes definitely only one acid, which is carbonic acid, since CO2 plus H2O equals to H2CO3, and H plus combines with deoxyhemoglobin to give HCO3 minus. H plus HCO3 minus, and this HCO3 minus can basically be expired through your lungs, therefore it's a volatile acid. The next one is non-volatile acids. Obviously we don't have carbonic acids only being produced in our body, we also have other acids being produced, which comes from metabolism, from phospholipids, from lactic acid, from sulfuric acid. How are these really excreted? These are excreted by your kidneys. Okay. Apart from the acids, I'll be talking about buffers. So we can divide buffers into two types. One is intracellular buffer and one is the extracellular buffer. So the intracellular buffers include proteins in your cell, the hemoglobin, which is deoxyhemoglobin, which is the strongest buffer in your body, in which the hydrogen ion, when carbonic acid is formed, binds to that deoxyhemoglobin. Okay? And the next one is the phosphate buffer, which is technically in your cells as well, because we have phosphate ions in your cell. Then the next one is extracellular buffers, which also includes ammonia and phosphate. The difference is that between which is a better buffer at the end of the day in the extracellular fluids. Obviously, we will think that ammonia and phosphate, they have a really good pKa, and therefore they are very prone to disturbances, they're prone to adjust in acid-base disturbances. However, biologically, it is the bicarbonate buffer which is more prevalent and more useful. There are only two reasons to this. One of that, since bicarbonate ions, when it combines with an acid, it can obviously be expired easily through the lungs. The second one is there's a lot of quantity of bicarbonate ions in our blood. Therefore, it's a better buffer. Coming back to ammonia and phosphate buffer. Ammonia buffer is produced in our body, technically kidney, whereas phosphate buffer is a titrable buffer, which is proportional to the amount of phosphate ions we have. So for instance, let me take a really good example, renal failure. In renal failure, we lose the ammonia buffer capacity. So what is the only buffer that is being acted upon? Phosphate ions. Okay. Coming back to acid-base systems which are involved. One of them is the red blood cell, or let's say blood for instance. The second one is the kidney. The third one is the lung. So I'll be talking about, it's always a combination between these three systems to really understand, and you need to really grasp this quite well. Okay. So let me draw a simple red blood cell. Okay. So our CO2 goes in, combines with H2O to form H2CO3 minus. What catalyzes this reaction is carbonic anhydrase. Okay. This carbonic anhydrase dissociates into two things. One is H plus ions, the other one is HCO3 minus ions. This HCO3 minus ions goes out of the cell with an exchange of Cl minus ions going in, chloride ions. What is the purpose of this chloride exchange? Could you just think for a moment and explain me? Pause it and think it. Obviously. So we do this exchange because if this chloride transport exchange did not exist, the equilibrium reaction wouldn't move in one direction, as we can see right now. If the H2CO3 minus ions remained in the red blood cell, CO2 would never be really combined, because at the end it's an equilibrium reaction. Okay? It's always the forward balances the backward reaction. Coming back to the next system, we have the kidney. The kidney involves the ammonia and phosphate. Where is ammonia coming from? An amino acid called glutamine. Where does glutamine come from? Muscle and liver. Phosphate, on the other hand, is just variable to your phosphate diet. Then lung. The lung, basically, when we have the lung is involved in excreting volatile acids such as carbonic acid. So if we increase the respiration rate, we excrete more acid in the air. The next thing I'm going to talk about is electrolyte disturbances in acid-base balance. So when we are talking about electrolyte disturbances in acid-base balance, we always consider two things: the disturbance between potassium and the disturbance between calcium. Okay, so our body has adapted physiologically to prevent hyperkalemia or hypokalemia. Our body has adapted physiologically in order to prevent extreme acidosis or alkalosis. However, it comes with serious adverse effects. It either causes a high potassium or a low potassium. So for instance, let's take, this is our cell. We have a lot of potassium ions, okay? For instance, if we have acidosis extracellularly, we need to prevent that acidosis. So what our body does is, it exchanges hydrogen ions in concurrent of potassium ions. So what happens to the potassium levels in the body? It causes hyperkalemia and acidosis. However, in alkalosis, it goes the reverse direction and it causes hypokalemia, because the hydrogen ions needs to be pumped out in order to maintain the normal body pH, which is between 7.37 to 7.42. Okay, the next one is what's the problem with calcium ions? Okay, so as we know, our proteins, our albumin are really good acid-base buffers. So there's always a competition between two ions. One is calcium and one is hydrogen. For instance, in acidosis, the hydrogen will displace competitively these calcium ions. So it will cause hypercalcemia. However, in alkalosis, it will, obviously, alkalosis, there's no hydrogen ions, so more calcium ions will bind. So it will cause hypocalcemia, thereby causing a very known phenomenon called tetany. Do you know why we have tetany? Obviously, calcium ions are involved in stabilizing the sodium channels in the muscles. So, since we lose the stabilization, we have increased influx of sodium ions, therefore increased contraction, increased action potential, and therefore tetany. Okay guys, so I will end my brief video for now, but I will continue with the acid-base disturbances and how kidney is extensively involved. I'll make another video because the kidney plays a really crucial role and it's quite complex. This is just a brief overview. Try to get this in your head. And yeah, that's it. If you have any questions and doubts, please comment below. Thank you.